Hello everyone. Today we are going to discuss about the autoimmunity. Development of immune reaction against the self antigen is known as autoimmunity. As we have already discussed in immune tolerance, that immune tolerance is responsible or immune tolerance prevent the development of immune reaction against the self antigens. Failure of immune tolerance will result into the development of autoimmune reaction. Autoimmunity or autoimmune disorders are principally because of two mechanisms. First is inheritance of susceptibility genes and the second is environmental triggers. In inheritance of susceptibility genes, they will result into the breakdown of self tolerance, and environmental factors like infection, chronic inflammation and various physical insult will result into the self-reactive lymphocyte. So in pathogenesis of autoimmunity, a role of susceptibility gene, they will result into the failure of self-tolerance and there will be increased self-reactive lymphocyte and they damage to self antigen or self tissue. In environmental triggers, infection, inflammation and tissue injury may result into the activation of various antigen presenting cells and they activate the lymphocyte, they become self-reactive and ultimately result into the tissue injury. There are certain abnormalities that appears to contribute the development of autoimmunity. First is defective tolerance or regulation. Defective immune tolerance and regulation may result into the development of autoimmunity. Second, abnormal display of self antigen. Increased expression of self antigen may result into the development of autoimmunity. Post-translational enzymatic modification of self antigen or various cellular stress or injury may result into the development of new antigen or new epitopes may also contribute to the development of autoimmune disorders. The third mechanism or third abnormality which also contribute to development of autoimmune disorder is inflammation or initial immune responses. They may also result into the development of autoimmunity. As we have discussed, autoimmunity is primarily because of the susceptibility genes and environmental triggers. In susceptibility genes, we have two classes. One is HLA alleles and the other is non-HLA genes associated with the autoimmune disease. In HLA alleles, there is example of HLA-B27 which is associated with the development of ankylosing spondylitis. In non-HLA genes, there are several genes, namely PTPN2, PTPN22, NOD2, interleukin interleukin 2 receptor or CD25. Mutation of PTPN22 will result into the defective functioning of tyrosine phosphatase and will result into the excessive lymphocyte activation. Mutation in a NOD2 gene will result into the defective cytoplasmic sensing of the microbe and result into the chronic inflammation, and chronic inflammation may result into the development of autoimmune diseases. Polymorphism in the gene encoding interleukin 2 receptor or CD25, they affect the balance between regulatory and effector T cell. So these are some examples of genes that involve in autoimmunity. Now we will discuss certain disorder associated with these genes. Disorder associated with HLA allele in autoimmune diseases. This table is from the Robbins or in which we will discuss about the diseases associated with HLA allele, like rheumatoid arthritis, type 1 diabetes, multiple sclerosis, systemic lupus erythematosus, ankylosing spondylitis, and the celiac disease. Sometimes question directly arise from their HLA allele or associated HLA allele. So this is a very important table for the NEET PG perspective. Another class of gene are non-HLA genes that are associated with the autoimmune diseases, like PTPN2 mutation associated with the rheumatoid arthritis, type 1 diabetes, and IBD, inflammatory bowel disease. Interleukin 23 receptor associated with the inflammatory bowel disease, psoriasis, and ankylosing spondylitis. CTLA4 associated with type 1 diabetes, rheumatoid arthritis, and interleukin 2 receptor associated with the multiple sclerosis and type 1 diabetes. These are the examples of non-HLA or non-MSC genes, in which genes involved in immune regulation. Now in MSC, non-MSC genes or non-HLA genes, there is a class of genes involved in immune response to microbes, like mutation of NOT2 gene will result into the development of IBD, and IBD there is also a mutation of ATG16 also noted. IRF5 mutation is seen in SLE, systemic lupus erythematosus. We have discussed about the susceptibility genes and their associated disorders. Now we will discuss about the roles of infections and the other environmental factors. Roles of infection in autoimmune diseases. The microbes incite autoimmune disease by two mechanisms. First is induction of the co-stimulator or antigen presenting cells. Microbes on exposure to antigen presenting cell will result into the increased expression of co-stimulatory molecules like V7 and result into the excessive activation of the T cell and ultimately result into the tissue damage. Another mechanism is molecular mimicry. Microbes resemble the self antigen and they result into the resemblance to self antigen. T cell activated against these microbes will result into the damage of the self antigen and result into tissue injury. There are other physical factors like UV rays will result into cell death. Upon cell death, releasing of the nuclear antigen, and these nuclear antigen incite immune responses, particularly in the lupus. Smoking may also result into the chemical modification in self antigen and development of autoimmune diseases. Some local tissue injury will result into the release of self antigen and there will be autoimmune reactions, particularly in immune privileged site like testes, brain and eye. Autoimmune diseases are usually more common in the female. Now we will discuss about the general features of autoimmune diseases. Normally they are chronic and multiple relapses and progressive in nature because of the epitope spreading. What happens? An immune reaction against self antigen will result into the tissue damage, and on tissue damage there is release of other or new antigens. They further activate lymphocyte and counter to new antigen, and this will result in a progressive disease. So this is epitope spreading. Autoimmune diseases and their clinical and pathologic manifestation depends on the nature of underlying immune response. Clinical and pathological manifestation of an autoimmune disease depends on or determined by nature of underlying immune response. If the excessive functioning of Th1 and Th17, this will result into this is seen in psoriasis, multiple sclerosis, and inflammatory bowel disease. Whenever there is involvement of antibodies or T cell mediated inflammation, this will result into the rheumatoid arthritis. Systemic autoimmune diseases usually involve blood vessels and connective tissue. They are also known as collagen vascular diseases or connective tissue diseases. Thank you very much.